Welcome back, everybody. Shall we get into the world of viruses? I think we should. But first, number one, I'm going to ask you guys a big question. What is the difference between bacteria and viruses? Like, what are viruses? I keep telling you they're not alive, but what are they? Do you guys know? Do you have any concept before coming to this class? Did you have any idea? What even, like, why are they a different thing? So I'm hoping that when you guys leave here, for those of you who have no idea even what that means, that at least you'll have an idea that when they're talking about COVID virus, you can understand why that's a different thing than like any bacteria or anything like that, and why you can't treat them with antibiotics and stuff like that. Okay, so all right, so what are we going to do in this chapter? We're going to talk about the sizes and shapes of virus, the components of a virus particle, the classification, the steps involved in virus replication, comparing the ways that we can cultivate or culture viruses, the challenges with viral infections, treating them, not antibiotics, not antibiotics. Antibiotics are for bacteria only. And then prions or prions and how they replicate. My dog is snoring and I apologize, but she's super old, so I don't really like to tell her not to do things. Okay, some names, right? We love the names. Can't not love the names. This is what you need to know as far as the names go, big picture wise for this chapter. There's of course other ones, you guys know this, but be familiar with these guys. Definitely Pasteur. He developed the vaccine for rabies and came up with that term virus, which he was calling a poison because at the time they didn't understand what viruses were. They couldn't look at them and figure out that they were their own little thing, but they knew that we'll get to it in a little bit. They knew that it wasn't bacteria. They knew that it wasn't what they were used to seeing, what they'd already discovered. Ivanovsky and Biernik, they discovered tobacco disease caused by virus. It was an important problem for tobacco crops. And then Loeffler and Frosch did the foot and mouth disease. They showed that was probably caused by a virus, right? So once we figure out what viruses are, we can kind of start discovering a little bit what's causing what, stuff that we didn't understand before. So okay, filterable. Viruses were filterable. So essentially you could take infectious fluids, whether it's urine, whether it's ascites, which is the fluid in the abdomen that can build up in some folks, whether it could be, diarrhea or whatever, blood, whatever it is, you can filter it and all the bacteria is removed. And in people who just had a bacterial infection, that should make whatever comes through the filter sterile on the other side. But that wasn't the case with these people who had viruses. We could still make people sick with that fluid. Right, so viruses can infect any old type of cell. If there is a cell, it can have its own virus, right? So we can infect bacteria, algae, fungi, protozoa, plants, and animals, all of them, right? And there's a lot of different sizes that can come in. We'll get into that. So what is the nature of a virus? Why are you barking? What is the nature of a virus? Are they organisms? Are they alive? No, they are not organisms and they're not alive. We already said with cell theory that in order to be considered alive, you had to have a cell that could divide and make its own replication on its own. Viruses cannot do that. They are essentially can just be protein coated nucleic acid, and they need a cell in order to replicate. They need a host cell, right? So what role did viruses play in the evolution of life? There's a lot of indication that a lot of our genome was probably just caused by viral infections that we didn't know about that got stuck in there forever. It's really, I'm not kidding. And that may have had a hand in how we evolved to be what we are today. True of a lot of organisms, right? Their distinctive characteristics, the differences between each of them. I mean, they are very different from one another. It's very, very small. Like we said, it can pass through these, super tiny filters. How can it cause so much disease and death? We'll get into that. The connection between viruses and cancer, oncoviruses, remember oncology, oncoviruses. We're going to get to those. And then what role did viruses play in the development of all other organisms. Same thing as with our own evolution thing. So we talk about them in terms of infectious particles rather than organisms. So this is what I was getting at. It's like, I'm sure like a lot of you would have gone to like, you know, Walmart or whatever, and you buy this and you don't think twice about it. It says kills 99% of viruses and bacteria, but you can't kill viruses. So what exactly is this talking about here? Are we talking about like, you know, so what we call that? So that's what this next part is. You would either call them active or inactive. If you can put them into another cell, a host cell, where they would have been happy and growing and make that cell sick. If you can still do that, then it's active. If you can't, then it's inactive. So Lysol's making a claim that you can inactivate the viruses. Obligate intracellular parasites. What does that mean? Obligate. Now, this is a term I really need you guys to get familiar and comfortable with. Obligate. I have said before, if it's obligate, you are obligated to do it, right? You have to. Mama said you need to take out the trash. Mama says you are. It's, you know, your obligation to take out the trash. I'm not going to tell you again. Obligate. It's their obligation to be intracellular. Okay. I don't know why people have a hard time with that term. I mean, I guess I do because it's just worded in a weird way. But yeah, so do what you need to do to help yourself understand whatever is going on with that term, right? We're going to use it a lot. Obligate. So obligate intracellular parasites. Viruses cannot multiply unless they invade a specific host cell. Some of them are very specific, and they must instruct the genetic and metabolic machinery of that host cell to make and release new viruses. That's pretty much why it has to be specific, because they can't control the cell if you know, they can't control the cell. You can't replicate. We're so loud, Hazel. Okay. So what makes a virus a virus? Well, how are they special? So they're not cells. They don't have cells. They never will be a cell, right? They're obligate intracellular parasites. They'll live in all these different other types of cells, prokaryotic or eukaryotic. Sorry, I wasn't expecting that to pop up. Right. So they don't independently fulfill characteristics of life. So technically not alive. Inactive molecules outside of the host cell and only active inside of host cells. They have a very basic structure, a protein shell, which we call a capsid. We'll get into that, surrounding a nucleic acid core. And I mean, they've had a huge impact on life. They're small, they have DNA or RNA, but not both. We have both, right? In our cells, inside of us. They will only have DNA or RNA. But not both, okay? And their genomes are ridiculous. They can be single-stranded DNA, they can be single-stranded RNA, they can be double-stranded DNA, like ours is, or double-stranded RNA, which is weird. They can be positive orientation or negative orientation. They can have stuff on the surface to allow for specific attachment to host cells. They multiply by taking control of host cell genetic material. They lack enzymes for most metabolic processes. Sometimes they steal from other cells. And then they lack machinery for synthesizing proteins. They don't even have their own ribosomes. They don't even have their own ribosomes, y'all. So here, this stuff in table 6.1 is a pretty good rundown of everything special about a virus. So viruses, are they alive? No, they're not. Not alive, pulses. Okay. There's sizes and how big and small these viruses are. They are super small. Some of them can be a little bit bigger than the smallest bacteria though. So just FYI, letting you know, right? But they're tiny. These are on the tiny scales, all right? These are electron micrographs. So some pictures of what they can look like. The one over here, which I think is kind of the cooler one, is Ebola. Ebola has what they call a shepherd's crook. But yeah, there you go. Right, so they don't even look like cells. They do, they can't even make their own proteins, but yet they're made out of nucleic acids surrounded by proteins. So where are they going to get all that? Well, from the host cell, they're going to make the host make it. So in order to get that stuff, they have to take over a host cell. They only need, for a virus, the bare minimum. And I want you to know this, okay? These two things. And this one here, external coating, that external coating, I'm gonna write it up here, is called a capsid. And we'll get into that, no worries. But you only need a capsid and then nucleic acids. That's it. It's pretty nuts when I tell you that our cells have like an endoplasmic reticulum and a Golgi apparatus and a mitochondria. And the mitochondria used to be a whole other cell in and of itself. And we have, let me tell you about the metaphases and all that stuff. No, not with the virus. So let's break them down, shall we? Virus particle. Okay, so we know that it has to have a covering of some kind. And we know that it has to have a central core. That's the nucleic acid, right? So that is our nucleic acid. And sometimes there's other proteins. I don't really want you guys to worry about this though, okay? But what I do want you to know is that central core, nucleic acid of some kind. It can be DNA or RNA, not both. Oh, why? No, sorry, I'm holding this right on that button. Okay, DNA or RNA. Okay, so then now we've got the capsid up here. So the capsid is the protein coating. It's the protein coat over the nucleic acid. And that is just a general term, nucleic acid. Nucleic acid, I'm just talking about DNA or RNA, right? That's all that really means. I promise I'll work on that. I'm just holding this the stupidest way you can imagine. Right, so that's that. Some of them, not all of them, also might have on as part of their coating an envelope. What does that mean? It just means a membrane, and it always steals the membrane from the host. Okay, so I'll show you a little bit what it looks like whenever they bud out, but yeah, that's the idea. So here are some general ideas of what they would look like. This is the one, weird. Okay, this is the one with just the capsid. There's no envelope. And then this one over here has the envelope. So you can see this pretty much has the same thing on the inside that's over here, right? So, but these are the spikes. I do want to point these out. These allow the virus to get into the host cell. They're very, very, very specific. And they're just a protein. And it's a glycoprotein, so often have sugar associated with it. Glyco means sugar, protein, okay? So they are the lock. They mimic usually another molecule that we normally see in our cells, like our cells normally interact with it. So they'll mimic another normal thing, come up and have that fake copy key to get into the lock to get inside of the cell. That's what's going on with the spike, glycoprotein. And so the spike, here's how you spell it. Almost clicked it again. Okay. Okay, so this is the general breakdown. We're going to get into all of this. So here, oh yeah, I should have just done it over here because it's all written here. So a virion, a single, just one particle, we would call a virion. Okay. Capsid equals the protein. So this is an important word. This will come up over and over again. Please be familiar with it. Become like second nature with that word. A protein shell surrounding the nucleic acid. The nucleic acid for this virus, we're only just talking about its genome. Okay, their nucleic acid. That's what kind of nucleic acid can they have? DNA or RNA in a bunch of different forms. Okay, we have repeating subunits called capsomeres. So I know it says capsomeres, right? So we have capso, and then I want you to notice this word, mer. So think of it that way. The word is capsomere, but when you say it, think capsomer, because we talked about polymers and monomers, right? And polymers is multi-mers made up of, right? So polymers made up of multiple single mers, right? Here, capsid, its parts are like capso, like capsid, mers. So when you see it, like what are the things that make up a capsid, the repeating subunits, and you would see capsomere, you can recognize it now because it makes total sense, right? Okay, nucleocapsids. Once we have the capsid, that protein coating around the nucleic acid, and it is actually around the nucleic acid, that unit together is called a nucleocapsid. And we're going to bring this word up a lot too. So nucleocapsid, that's nucleic acid with a capsid. That's all that is. Some viruses, that's all you have. Other ones can have an envelope. A lot of them do. So that's just the membrane it steals from the host. And the spikes, it's a glycoprotein on the outside to allow it to get in and out of cells, not out, to get into host cells, right? So it allows viruses to, we'll say dock. That's a pretty good word for it, dock. Okay. So just looking at a bigger picture of those, we've already gone through these. I'm not going to go through them again. Capsomeres, those subunits of the capsid, right? They're identical. They spontaneously self-assemble. That's because of the chemical interactions between those units. So they want to come together and work together to create the capsid. Okay. There's two kinds of capsids you can have. Helical, really what it sounds like, literally a helix going down, or icosahedral. Do you guys remember what that word is? Icosahedral. An icosahedron is a shape that's made up of repeating isosceles triangles. So it's just isosceles triangles over and over again. Okay. That's what we were just looking at in this other picture. Right, you see the triangles. This is an icosahedron here. Yeah, and it is just like this. Viruses are nuts. Okay. Yeah. Okay. So here's the helical version. You get these little units, these subunits. So these are, where's my guy? There it is. The capsomeres, each one of these little pill guys, that's a capsomere. They're coming together in a helical way to form the capsid, and the nucleic acid is going to come sit in the middle of it, right? So this is essentially what it's going to look like in the end. We've got the capsid on the outside and the nucleic acid on the inside. Down here, you can see it kind of still going with that helix coming up, that helical, and it's going to keep going and going and going and going, whatever. And then you can pop this sucker, this whole thing. So we got this rod. So some of them, that's all you need. There's a rod of a virus and it's got a helical capsid. That's it. It's happy. Some of them you can take whatever, a similar thing to this protein coated nucleic acid that looks a lot like that. And then you can like just let that flop on down like a telephone cord, or you can let it flop on down like a little snake, or shove it in there just all random like or whatever. But essentially, you shove it in to a envelope. You have that whole thing come into, get pushed into that envelope that's studded with those spikes. Okay, so that's the envelope version of the helical one. Okay, these are the isosceles triangle ones. I mean, they're pretty straightforward. You've got your capsomeres. Your capsomeres come together in, I don't know if it's my, yeah, it is. It's my bracelet. I keep wondering what it's making it go weird. Oh, whoa. Okay, so you got the capsomeres that come together essentially in almost the shape of a triangle. And then those triangles will come together and create that icosahedron. That's the idea there. That can be it with the spikes on the outside, or it could be enveloped. We can shove that whole thing into an envelope and put spikes on the outside of it. So I mean, that's the idea anyway. So I mean, this pictures of that, and especially when they can have a pretty specific shape with that icosahedron going on, but it can sometimes be a little bit harder to see it if you don't have like, I mean, it's organic, right? It's not perfect. This though, this is insanity. This is a virus, y'all. This ain't a bug. This is a virus. So this is just protein and nucleic acid. That is all that is. I promise. So this is a bacteriophage. It's a very, very complex capsid. Okay, so bacteriophages are bacteria. No, bacteria infecting viruses. That's what I'm trying to say. So those opposite wording of what this was on here threw me off. But bacteriophages are phages, which is another term for a virus that infects. We'll just use the shortened version. Viruses that infect bacteria. Okay, so bacteriophage or just phage. Okay, bacteriophage or phage. Viruses that infect only bacteria. Bacteria are only infected by phages. Okay. So Multiple types of proteins going on here, obviously, because they're all going to interact with each other in their specific ways and then come together to have their function, right? But yeah, it's pretty crazy. Essentially, you're going to have the caps, the normal virus on the top over here. This is what we normally would think of, right? The nucleic acid covered by a capsid, right? But now we've added on this thing, which is a collar, and this thing, which is the sheath, which is sort of like a spring, these tail fibers that allow for docking with the cell so they can recognize it correctly. These are tail pins, which literally are going to help penetrate the membrane of the bacterium, right? It comes in, it recognizes with its little feet. When it does that, there's a chemical change that happens that causes the little legs to bend downward so that the tail pins can stick in. And when they do, there's a chemical change within the sheath that causes it to inject the nucleic acid into the bacteria. Literally, that's how it works. Okay, so moving on, different forms of the capsids and what it can look like. I mean, there's a lot of different versions. This one down here, I will tell you, is HIV. It has a very unique shape to its capsid. But it's also got an envelope. You can see that's what that purple is on the outside, and the teal is the spikes. Okay, so protein shell that surrounds a genome is called, do you guys remember? Capsid. Okay, the envelope, that membrane coating that some of the viruses have, it's always from the host. I'm sorry that this is in the way here. Oh, what? I don't even know what that was. I don't even know what that was. Okay. All right, the viral envelope is composed of the membrane system of the host. We have, it can be from the cell membrane, like the outside of the cell, or it can be from the nuclear membrane, if we're talking about eukaryotic cells, right? So it can steal it from that. So, right, so it's going to also make so much of its viral proteins that on the outside of that membrane of whatever cell it's infecting, instead of having its normal proteins of its normal function for that cell, it's going to be studded only with spikes so that those things can bud out. The virus's new viral particles can bud out and have just spikes, happy, ready to go on their surface that aren't interfered with any of the normal cell proteins. It's kind of gross and scary, but yes, that's how it works. Okay. What is the lipid membrane that surrounds some viruses called? The lipid membrane that surrounds is the envelope. So that's right, that membrane is always a lipid bilayer. All right, getting into it, nucleic acids, whatever their genome is, okay? Viruses, their genome is whatever is included in their DNA and RNA, whichever one they have. All right, so I guess this is wording it weird, I apologize. For cells, for actual living beings like you and I, or the bacterial cells, or protozoal cells, or whatever it is, protozoan cells, or whatever it is, those cells, their genome is all of the DNA and RNA and everything included in it. Their chromosomes are their genetic information that's, passed down genetically, like speaking, okay? But yeah, for viruses, you will have either DNA or RNA, but not both. And they're not typically functioning in the virus. They're not functioning until they get into the host cell, okay? I don't know if we're coming back to this picture, so I do want to touch on it a little bit, but you can see here we have a double helix. We have a double helix of DNA. That's like a normal, like double helix. You guys are used to this, right? That's not a weird thing to you. Our DNA, our genome exists as a double helix, 2 strands, and they're complementary to one another. And they actually run technically in opposite directions, depending on like the chemical makeup of them. But like one is the exact opposite of the other, but you can still get the copy of the other one from it if you just make the complement of it. So that's how it's useful. But anyways, there's 2 strands, the positive and the negative. The normal one that contains a message and the one that you can make a copy of to contain the message. So positive and negative, that's how the double helix is. So that's why this thing is talking about positive versus negative. Okay, so that positive one being the one that actually carried the message, and the negative one, you'd have to make a copy of it for it to have carried the message. Okay. God, my back hurts. So yeah, but that's the idea there. And then mRNA always is positive orientation. Whoa. mRNA is always the positive orientation. So you'd always, if you had a positive genome, you would have to put negative in order to make positive mRNAs from it, for example, or make a negative in order to make positive copies to put in your new viruses. So stuff like that. Okay. But they can have any of them. I don't know who that was. But yeah, okay, here's just the types of nucleic acids that viruses can have. I'm not going to ask you about these, but just to start, like if you go to the top, we have a very large virus, the variola virus. It's the cause of smallpox, and it has double-stranded DNA. A lot of the bigger ones can have the double-stranded DNA. We go down all the way to the bottom, we have HIV. HIV has a single-stranded RNA. It also has reverse transcriptase. What does that mean? That means that reverse transcriptase is an enzyme, which is a protein with a function that allows that virus to put its RNA genome into a DNA form, and that DNA form can be put into the host cell's genome permanently. Okay. Okay. Other things they can have inside of their virus. We talked about the capsid, we talked about the envelope, and it has the spikes, right? And we talked about the nucleic acid. What else can they have? They can have polymerases with them. They can have replicases to make copies of the RNA. Polymerases are what make actual new synthase. So polymerases make actual new nucleic acid. Okay, so nucleic acid particles. It's like DNA or RNA strands. The replicases are just going to copy the RNA. The reverse transcriptase is going to turn RNA into DNA. Substances from the host cell, they can also package up ribosomes with them to take them to wherever they're going next. And they can also borrow tRNAs, and tRNAs are tRNAs are molecules that are going to allow the cell to carry amino acids. So remember how I told you about there's an mRNA messenger that's going to say these are the instructions for making the bedroom portion of the house, right? We've made a copy of just the bedroom gene. So that's the messenger RNA. The ribosomes come and sit on it. And when I say that the ribosomes read it in 3-unit pieces, 3 nucleotide pieces, what I mean is they are like a car, and the tRNA, the transfer RNA, come in and recognize the correct sequence of those 3 units. And then when it's correct, that amino acid will stick on to the protein that it's making. So that's what tRNAs are. So some viruses steal that. So how do we classify them? We might classify them based on what they can infect, whether they're DNA or RNA, whether they're helical or icosahedral, their structure, chemical composition, the genetic makeup, all of that stuff, right? Pretty obvious stuff. So this is just a classification of viruses. We're going to start over here on the left, which is showing, I did not even think this through. The order is the biggest, and the most specific is the species, okay? And over here, we have like whatever host type of organism it could infect, right? So we're going to go through this, not each one, but the idea of how it's organized, right? The families are all going to end in viridae. The genera are all going to end in virus. Okay, and then we have some that are called nucleocytoplasmic large DNA viruses. What does that mean? It's not so important to you guys, but they all take up some cytoplasm inside of their cells as well. But they're very large DNA viruses. They're all related to each other, but really not at all to the other ones. So they might be their own order. But yeah, so this is just looking at the DNA viruses first, and whether they're double-stranded or single-stranded. We talked about what that means, double-stranded and single-stranded, right? So whether they're double-stranded or single-stranded DNA, then whether or not they're RNA, whether they have the positive polarity, which actually is the message, or the negative polarity, which you use to make the message, right? Double-stranded RNA, which is unusual, and then special RNA, like the ones that with the reverse transcriptase that go from RNA to DNA. So that's what this table is showing you. If you want to go through and see which ones are which and be interested in that, that's all in there. This is those genomes, and this is showing that DNA double helix here on the right so that you guys can see a C is always opposite of a G and a T is always opposite of an A in DNA, only in DNA. RNA has the U in place of the T. But we will go over the details of that stuff in the next unit. But this is just to show you guys so you can get a sense of this is what the double helix would look like. One side is the positive, and on one side is the positive, reading the actual message. The other is the exact complement of opposite bases that you would use to make that message, right? So it's just the opposite of it. So you can split them apart and you'd have a positive and a negative. They'd be single strands, right? That's the idea with that. And then with RNA, they're just talking about doing it with the RNA parts instead of the DNA parts. Okay, I just want you guys to be aware of that. It's hard to wrap your heads around, but there you go. Okay, how they're classified and named. Mostly structure, chemical composition, and genetics. Okay, mostly. So then we have the a realm, which is the virias, all viruses. The orders, they'll all fall into 6 of them. That ends in virales. Viridae is the families. They'll sometimes have subfamilies called Virinae. And then the genera is virus. And then there's the species, which you would just name them whatever that virus is, like human immunodeficiency virus or something, right? So this is how they name the actual virus species. Sometimes they name it after the disease. We have foot and mouth disease virus, Ebola hemorrhagic fever virus. It's pretty straightforward, like we just did with HIV. And then we also have ones that are named after the place, like West Nile virus. But then we also have ones that are like a combination, like Crimean Congo hemorrhagic fever virus. Okay, Crimea is like near Turkey. Congo is in Africa. You guys know that. Okay. Multiplication cycles. Definitely know the order of these. 100%. Okay. So the phases of life cycle in infection with a virus. Adsorption with a D. Penetration, uncoating, synthesis, assembly, and release. We're going to go through each of these. This is going over it for the RNA viruses. And then there is also one for the DNA. Really quick, all I'm going to do is run through these. Adsorption, sticking on to the surface. So that is number 1 over here. Just our spikes. Remember I told you they're like going to dock or be like the copy key to the lock on the surface of the cell, right? Mimics a normal protein that would be interacting with the cell surface. So that's adsorption, sticking on. Okay, penetration. The cell is now engulfing it. As a result, that's what it wants. Then we have uncoating. That's going on right here. Literally the cell's breaking it down so that the genome can come out, or it'll just, like, as a reaction of being inside of the cell, the chemical environment will cause it to release its genome. Now we have the genome, which in this case is a positive RNA for this organism, not alive, this virus. It's going to release positive RNA. So it'll make a negative copy so that it can make a bunch of positives, right? Or we can actually literally read off of this one to like an mRNA messenger, right? With our ribosomes and stuff from the cell that we're hijacking to make capsomeres and spikes. Okay, so that part is called, it's number 4, that's synthesis, replication, and protein production, right? Number 5, we have assembly. Well, that's pretty straightforward. I don't think I need to describe it, right? You're putting the capsomeres around the genetic new genomes that you just made over here, putting these with this and studying them with that, right? Well, in this case, we have an enveloped virus. So I'm sorry. So let me back up a little bit. We're going to put the capsomeres around the nucleic acid. The spikes are going to go to the surface. That's all that's going to be on the surface. We're going to knock off all of the normal proteins that would be there on that cell, all of them, and replace them completely with these spikes. So that when we make up that capsomere, the capsomere crap around the genome, then that is a unit Do you remember the word for that? It's just when I get really low on this tablet that I start having this problem. My hand doesn't sit on it right. Nucleocapsid. Okay, or nucleocapsid, whatever works in your brain. It's not wrong to say it either way, but yeah, so that part is just the nucleocapsid. And then it's going to bud out and cover itself with the envelope by stealing that host cell membrane and putting it around itself and budding out. It's called budding. Okay, so in this, that's the release. That's number 6. Okay, this is the DNA ones. And the DNA ones, essentially what we're going to have is the DNA virus can release its genome, whatever. It's going to take it into the nucleus here. And then it's going to allow for production of this yellow thing. Now, what is that? That is mRNA. That's our messenger of whatever proteins we need made. So whatever viral proteins are over here. So it could be the it could be the capsimeres, or it could be the spikes, that sort of thing, right? Also, we can make more DNA. Also, we're broken it apart into single strands and we'll make copies of the DNA. So we've got replicated DNA to put into the new virus parts, right? Cool and everything, but it can also put that into the host DNA, the new double-stranded DNA. Okay, so now we've got all of these proteins that got made outside of the nucleus and got shuttled back in. We've got our capsimeres and our spikes and whatever, and they're coming together around our DNA to make the mature virus. In this case, we have a virus that has not got an envelope, but you can also make an envelope around these. You would just, because you're already in the nucleus, you would just bud through, do what we did on the outside of that cell. Oh God. Through the actual nuclear membrane instead. Okay, now we're moving on. Adsorption, starting with that. Sticking to the outside. Adsorption means sticking to the outside, attaching on the outside. Absorption with a B is like soaking the thing up with like a paper towel. Host range is whatever that virus can infect. Some of them have a lot more specific host range than others, and it's all going to be mostly determined by those spikes is what we're talking about, okay? So what those keys fit exactly into that specific thing. So we have a restricted, which is the specific ones. Those are like, we have the example that is given here. We have hepatitis B. Only affects liver cells of humans. Can't infect any other cell type or any other animal, their liver. No, only human liver. Okay, then we have the ones that are somewhat restrictive. So the moderately ones. Here we have poliovirus. It infects intestinal and nerve cells. This already got a little bit of variation. And that's of all primates. Includes humans and chimpanzees and stuff, right? And then we've got broad, ones with a broad range that pretty much infect like a lot of stuff, right? Rabies virus. So rabies is the worst, okay? You think it's not so bad because it's been controlled. You are wrong. It is like the worst. It's terrifying. They make all these like zombie movies about it because they think that's like how to make it terrifying. But no, it doesn't need help. Believe me, it doesn't need help. Be glad for the rabies vaccine. Old Yeller is not even the tip of the iceberg. So I'm just saying, various cells, we say so general, that's just various cells, a lot of different kinds of cells for all mammals, all of them, including you. Penetration and uncoating, endocytosis. Endo, when you see that, I want you guys to think inside. So that could be taking something inside or whatever, but inside is the general thing. Cyto means cell. Cis, just think of it as like it is taking it inside the cell. That's the action that is going on, okay? Endocytosis. So the cell is gonna like literally come up and be like whoop, around it and take it in, okay? Eats it like a mouth. So we'll take it into a little vesicle that way. And then that usually will release certain enzymes into the vesicle that break down the virus, right? So here we have a virus that is getting taken up like this. See the engulfment? And then that virus is in a vesicle and it'll start getting broken down. Really. And then we'll have our free DNA into the cell so that it can do its nasty business. So here we have down here underneath it, an enveloped virus. So that membrane is that blue, like darker blue here. So it's going to come up, it's going to interact with its surface thing, and then it's just going to literally fuse with that membrane. So that's how it's going to do its uncoating. It's just going to fuse with the membrane. It doesn't need to be broken down in a vesicle. Okay. Okay, so that's uncoating is just taking off the capsid and envelopes so that we're releasing the nucleic acid. Let's do it.
There are two mechanisms by which enveloped viruses enter host cells. In one of the mechanisms, the virion attaches to host cell receptors by specific proteins on its surface, called spikes. The envelope of the virus fuses with the plasma membrane of the host, and the nucleocapsid is released directly into the cytoplasm. The nucleic acid then separates from the protein coat. In the second mechanism, the enveloped virus adsorbs to the host cell by specific proteins on its surface, and the virion is taken in by endocytosis. In this process, the host cell plasma membrane surrounds the whole virion and forms a vesicle. The envelope of the virion then fuses with the plasma membrane of the vesicle, and the nucleocapsid is released into the host cytoplasm. The capsid protein is then removed, releasing the nucleic acid of the virus. A naked virion also enters by endocytosis. Since the virus has no envelope, it cannot fuse with the plasma membrane. After being engulfed, the viral nucleic acid is released from the endocytic vesicle. The nucleic acid then separates from the capsid.
Okay. Okay, moving on to synthesis. This is replication and protein production. So we have to make a lot more of our nucleic acid and produce a lot of protein to make new virions, right? So the virus takes over the entire host synthetic and metabolic machinery in order to make its new virions. So if we have RNA, like we had just shown in that other one where I explained the difference of how they like do their different steps, the RNA is going to replicate in the cytoplasm that's outside of the nucleus, and the DNA is going to replicate inside the nucleus. So this is the thing that I had explained to you guys before. I'm not going to go through it again, but this was the difference. So the RNA inside the nucleus, which is the blue area, and the DNA inside that purple area. So where the actual like synthesis is going on. So when we make up viruses, like when we're talking about animal viruses getting infected, this is inside of a nucleus of an animal cell, and this is the nucleus area inside of the cell. And in here, you can see like we have viral particles that are forming, and these are all viral proteins that have crystallized, basically like lined up with one another over in repeating units, taken over almost that entire nucleus with just viral manufacturing. So how do we get the mature viruses, the fully made viruses out of the cell? So it could be by budding, which is kind of what I was telling you guys, coating their surface with that spike area. So here we have the nucleocapsid, which is, it looks like a little snake guy here, but it's the nucleic acid covered in the capsid proteins. Then the spikes are on the surface of the cell, and then the nucleocapsid is going to come and attach to the underside of it, and then it's just going to pinch in and then release the particle. So that's budding. Okay. Let me write it over here. And then we have exocytosis. So exocytosis is like, it's hard for me to describe it without a picture. Let me see, is the next slide has one. No, it doesn't, of course not. But basically, the cell is going to have, like in the Golgi apparatus, right? Remember how I told you it like makes lysosomes and vacuoles and like helps secrete things and all that. The viruses can get secreted that way too. So that is a form of exocytosis. So exo, remember I had described you, endo meant into the cell. Exo is going to mean out of the cell, cyto, out of the cell action, okay? Right, so the important thing to look at here, I want you to make note of this, that in the case of like most eukaryotic viruses, these viruses are going to be shed gradually without destruction of the cell. So they're just going to get budded out through this method over here, just kind of gradually. Whereas with bacteria, they just pop out. Okay. Ooh, geez. Okay, damaging the cell. Sometimes we can have some damage because of all this. I mean, you saw all those proteins that one virus was causing that cell to make. I mean, it was a lot for a cell to handle. So when it causes damages to the cell, we call those cytopathic effects. So cyto, remember, cell, pathic, like pathy means bad, like you got some bad going on. It's not bad, but you know, like you got an illness or disease of some kind. It's not always infectious, but pathy is bad thing, right? So cytopathic effects is bad for the cell. It's going to alter its appearance. It's going to undergo changes like with its size, its shape. It'll round up. It won't attach to surfaces or to other cells anymore. They could form inclusion bodies, which means they're little like vesicles that are filled with like just crap from the virus or like dead organelles that are associated with it or damaged stuff of whatever's going on. Then we have formation of syncytia, which is the fusion together of multiple host cells, like all at once. You get a lot of nuclei all together. So this is syncytia here on the left. You can see that, like the darker things, those are nuclei here in one cell. So, and then on the right, you see inclusion bodies. I mean, here's a table of viruses and what sort of cytopathic effects we know that they cause. I'm not too worried about you guys knowing that. All right, persistent infections, what's going to go on there? We have this thing. Okay, so some infections can lead to a carrier relationship. Have any of you ever had a cold sore? If you've had a cold sore, that's caused by herpes virus 1. And it can sometimes come out whenever you get stressed, right? You can get cold sores on the lip and whatever in response to that. Well, it's incorporated its own genome into your DNA, and that's how it's hiding out. So it crawls up into the nerves and it hides out up in the cells, up in the nerves. And then whenever you're stressed out and your immune system is not doing as well, then it'll come out and cause that. So that's a very good example of that. We're also talking about like shingles, right? So we know people that like later in life, they've had chickenpox. Later on in their life, if they don't get a booster shot to keep their immune system strong against that virus, they will get shingles. It's the same virus, what caused their chickenpox when they were a little kid. Varicella zoster. Okay, right, so when that is happening, we can develop what is called a provirus, and that is when the viral DNA is incorporated into the DNA of the host. That's what I was talking about with the herpes virus, right? It put its DNA into your DNA, or I'm not accusing you of that, but like, you know, the person's DNA. Then we can develop what is called a chronic latent state. So that just means it's not active for a long time. Then we have viruses that cause cancer. These are called oncogenic viruses. When we think of this, I want you guys to think of like oncology. And then, so onco means cancer. Can't see the thing. Genic, think of that as meaning it generates. Okay, so it's cancer generating. 20% of cancers are probably caused by viruses. Some of them we probably haven't diagnosed what virus causes that cancer. Transformation is the process by which viral genes that can directly cause cancer can affect your cells, right? So, and cause the cancer. So your cells can become transformed by that virus. And its proteins can cause loss of growth regulation. So remember all those checkpoints that we had in that growth cycle with the PMAT and all of that, the G1 and those, it can cause problems with those. So I'm not going to go through these specific ones down here, but if you're interested in it, there's some down here, the retroviruses, and then DNA tumor viruses, I want to think about HPV. That's a pretty good example of these. The cervical cancer. Okay, bacteriophages. What are bacteriophages, right? Viruses that infect bacteria. Yes. So bacteriophages, when were they discovered? Frederick Tort and Felix de Herel. I don't know how to say that correctly, but anyways, they discovered them. And they were we found that over time that there are bacteriophages, viruses that infect bacteria, for every known bacterial species so far. They can often give the bacteria genes that make them worse for us, more pathogenic. Pathogen is a disease-causing organism, so worse. So essentially what we have is just like what we were talking about before. We'll have a virus. This is a bacteriophage, right? Bacteriophage. It attaches. That's an absorption. Number two, it's penetration. It injects its DNA in, right? Remember how I told you about it, like squats down and literally squirts in the DNA. Never ever goes in there itself. The capsid never enters the cell for bacteria. Okay. So that's how the genome gets in there. Now, if we're going to like become incorporated into the bacteria's life forever, well, not forever, but for, indeterminate amount of time, we can enter what's called a lysogenic state. So how I remember this is I don't know. I always think of lyso as being like trying to lyse or break something open, but so I don't like that. So here I want you to focus on the genic. Okay, you have lysogenic versus lytic. Okay, so lytic, do think of breaking open. And lysogenic, think of the gene part. Okay, so we are going to literally take the blue genome of that virus and put its blue little genome into the purple genome of the bacteria, literally, incorporate it into that DNA so that when the bacteria replicates, it's also replicating that viral DNA. So bacteria replicate pretty quickly, right? I worked at a food testing place where we would prepare the meat patties from McDonald's in the morning at like early. It was like 7, I think, and 7 a.m. And then come around like 5 p.m. we were ready to test them. They were fully grown. That's crazy for E. coli. So it's crazy, right? E. coli has very fast doubling rate, but yeah, so they grow up pretty quick, but then you have doubled all of the viruses that they have too. So that's something to think about, and then it can come back out again when the conditions are favorable, whenever it says that it likes it, that like whatever's going on, get triggered by the bacteria. Oh, we're in a happier environment. Now I want to go ahead and do this, whatever. The cell receives a signal, whatever. Now the genome is going to come back out and we're going to make copies. So now we can continue on with whatever's going on, right? So we make all these copies, we'll duplicate all the genomes and all of the proteins involved in building new things. We'll assemble the new virions and they'll mature, they'll be fully assembled and fully ready to go. And once there's enough, they will just, for bacteriophages, they will release by literally busting out of the cell, literally. Okay, here's a picture of that. That's crazy. Viruses are crazy, y'all. So here is the phage life cycle. I mean, we can watch this, I guess. I don't know how long it is. All right, Hazel.
The life cycle of bacteriophage T2 begins with a bacteriophage particle binding to the surface of the bacterial cell. The phage particle injects its genetic material, or DNA, carried in the capsid of the bacteriophage into the host cell. Once inside the cytoplasm, genes in the phage DNA direct the degradation of the host cell DNA and are able to utilize proteins within the host cell for the synthesis of new T2 phages. First, many copies of the phage DNA are made. The phage DNA encodes the proteins which form the capsid and the regulatory proteins which direct their production and assembly into phage coats. The newly made capsid proteins and phage DNA molecules assemble into a new generation of phage particles, and the cell is lysed, releasing the mature phage particles.
All right, so you can be in the lytic phase or lytic cycle, same thing. That's the active life. You're making active life cycle of the bacteriophage. You're making new viral particles, and they're going to bust out and like a normal life cycle of what we typically think of with a virus. Then you can have the lysogenic cycle or lysogenic phase, where we're incorporating the genome of the virus into the genome of the bacteria. Okay, so those phages that are capable of becoming lysogenic, the ones that can do that, we call them temperate phages. Okay, so that's the word for those types of phages. They are temperate. They can undergo lysogeny. Okay, they can incorporate their genome. When the genome is incorporated and is inactive, the DNA of the virus is in the DNA of the bacteria. When the DNA of the virus is in there, we call it the prophage, that viral DNA, okay? And then we can induce it to come out, or it's induction or activation, same thing. Those are some terms associated with lysogeny. Be familiar with them. So we're gonna go watch this video, I guess, of lysogeny really quick.
When phage lambda infects E. coli, either the lytic or the lysogenic cycle may be followed. In both cases, the first step involves the phage attaching to the host cell and injecting its DNA into the host cell. In the lytic cycle, phage nucleic acid is replicated and phage genes are expressed, resulting in production of phage proteins. Mature phage particles assemble and the host cell lyses, releasing the phage particles. In the lysogenic cycle, the phage DNA is not replicated or transcribed. Instead, the phage DNA integrates into the host cell genome. The host cell can then replicate, carrying the integrated phage genome. The integrated DNA is referred to as prophage DNA, and the host cells carrying the prophage DNA are said to be in the lysogenic state. When the cells are exposed to ultraviolet light or to certain chemicals, phage induction occurs. The prophage DNA is excised and the phage enters the lytic cycle.
You're pretty cute, guys. All right. Like how they cuddle together. So we can have that process of lysogeny in humans, in bacteria that infect humans. That's what I'm trying to say. So not in humans. That is it's a different thing when that happens in humans. When lysogeny happens to bacteria, and those bacteria can infect people, sometimes those bacteria cause a worse disease because of the genes transferred to the bacteria from the phage. Okay, yes. So that's what I'm trying to say. So that would be, that's what we call lysogenic conversion. Whenever the bacteria get their new pathogenic trait, their harmful to us trait from their phage infection. All right, so that includes diseases like diphtheria, cholera, and botulism. They're all transferred by a lysogenic conversion by phage infection. Okay. So these are comparisons of bacteriophage life cycle versus animal virus life cycle. I would look through this table and go through those comparisons if I were you guys. I'm not going to review it right now because we're running out of time. How do we cultivate and grow and research viruses? So we have in vivo methods where we would infect actual animals and embryonic bird tissue, which is extremely important. As well as in vitro. So that's like tissue culture, right? So we have cells that aren't part of an actual living thing anymore. Why would we want to do any of this? We need to grow it up to develop vaccines. We need a lot of virus for that. We need to grow it up so we can do genetic analyses to learn more about the virus so that we can develop drugs against specific things dealing with the virus, making mutations in the virus to see if, oh, well, if we make a mutation in this virus, it's no longer harmful, but we can still generate antibodies against it. Vaccine, you know what I mean? So that's the idea there. You might want to actually do stuff like that. It's not all bad. What animals do they use for inoculation? I'm not going to go over, but it might be something just to be good for, just to be aware of, so you know what's actually going on with regards to studying viruses as far as inoculating actual living animals and how and where on their bodies they might be getting inoculated with whatever virus. So, all right, bird embryos are important, especially like when we talk about the flu virus. Embryonic development occurs within that protective shell, so we can keep everything pretty much confined and contained, right? So, and it's self-supporting, it's got its own nutrients in there. We've got living tissue for the virus to replicate in. So it's super useful for studying viruses. Cell culturing, we'll talk about tissue culture. That's an in vitro. In vivo means living. So in vitro, just think that not in a living thing, okay? We can use tissue culture. We use the cells of like what we call transformed cells from whatever source that grow pretty much uncontrollably, can grow forever, indefinitely. So we grow those up and can infect them with virus. So we can analyze however they grow and grow up a lot of virus to study their genomes and stuff like that. Most of virology's work will be done in tissue culture. Mine was. So down here in the lower right, you can see this is what a typical virologist would use for growing up these flasks. And I used a lot of the flasks, and I used a lot of the little plates as well for my immunofluorescence stuff. But like, that's not the point. But that's how they grow up the tissue culture. So their primary culture is from fresh animal tissue at some point. They grow it up. They grow it in a mitotic. They grow it up in a monolayer via mitotic division. So the cells are just dividing from themselves, and they will have whatever the original tissue traits have. So that's a primary cell culture. Okay. Then we have continuous cell culture, which is from an immortal cell line. And we had a primary culture from that originally. Usually this is from somebody that had cancer, or like some cell line that was treated with a thing to transform it, to turn it into immortal. Usually this is usually similar to cancerous, but they often have altered chromosome numbers. They're just not, their checkpoints aren't working, but that is useful for us in this setting. So we can have them grow rapidly and forever. And then that's where we get into the topic of Henrietta Lacks. She was a woman who had cervical cancer that had a sample taken from her, and it was used to grow up an immortal cell line. And it was done against her will, but apparently back then that was just how it was done. They didn't need a woman's permission. It was a woman. Why would they need your permission as a woman? They just take stuff like that. So they have a, immortalized cell line created from her cervical cancer cells. It's probably one of the most used cell lines in science today. I used the HeLa cells, HE from Henrietta, LA from Lax, HeLa cells, what we call them. So very, very, very useful cell line. Another useful one would be 293 T cells. Those are what they work with as far as developing the COVID vaccine. And I know they are human embryonic kidney cells, but let me tell you about that for those of you who are uneducated about it. Those cells were not isolated recently. They're immortalized. So we're talking about cells that were isolated a very long time ago and were immortalized, and people have just been using them and using them and using them. And at least some good is coming from what may have been, not necessarily the smartest decision, or maybe not the smartest, most ethical decision back in the day when that originally happened. But now we have this tool, we should be using it. We shouldn't just throw it away just because someone back in the day. That's my personal opinion, but You got to see the good that's come out of it. There's been a lot of research done with those cells. I used 293T cells. They're probably, I mean, just as useful as HeLa cells. So I don't know. You just got to think about how you think about that. Which one's worse? I don't know. Okay, so challenges. You're working with viruses, you're trying to find cures or treatments or vaccines or whatever. Viruses can have high mortality rates, so you don't have people really necessarily to work with. Drugs can be difficult to develop because if you're trying to target the virus, you're also targeting the process in the cell, because like if they're stealing the cell's machinery and you're targeting whatever the virus is doing, you're targeting whatever the cell is doing because it needed that machinery too. So antibiotics do not treat viruses. They do not treat viruses. Antibiotics can treat secondary infections by bacteria associated with viral infections. Like if you have a cold and you go to the doctor, they give you a antibiotic. They're not giving it to you because of the viral cold infection. They're giving it to you to prevent the sinus infection caused by the bacteria. That's what they're giving it for. So just, be aware of the difference there. You do not and cannot treat viral infections with antibiotics. They just work. They don't work that way. They don't work. So, so that's why they prefer with viral infections, vaccination is far more preferred because there can be such bad side effects with viral treatments. So, viruses also have high mutation rate because they replicate so quickly and so fast and so much. They can, have a lot of mistakes coming up, and some of them are very advantageous, so they can escape the drug or escape the vaccine very quickly. So we know that. What other things can cause diseases that are not cellular? So other non-living things that cause diseases. We're going to talk about prions. Prions. Okay, so the disease is called spongiform encephalopathies, literally sponge. I just wish I could see the thing a little bit. Sponge. And then encephalo. That's brain. So bad things going on with your brain, making your brain sponge-like, literally. Okay. Brain tissue was removed from animals that were having the symptoms associated with this, and it resembled a sponge. We'll talk about in more detail in Unit 4, but prions are usually associated with spongiform encephalopathies. Okay. They have like distinct protein deposits in the brains that are causing like the sponge thing. So the protein's not behaving the way that it should. In humans, we have a disease called Creutzfeldt-Jakob disease. It's a spongiform encephalopathy. It affects the central nervous system, gradual degeneration of health. It's transmissible, but it's not entirely proven how. We know that if you eat meat that has mad cow disease, that you can get what they call a variant form of CJD, right? But other ways, we're not entirely sure exactly all the ways, right? Then we have animals that are that have other diseases similar to this. Sheep have scrapie, mink have a form, elk have a form, and then we have bovine spongiform encephalopathy, where we're talking about cows. So that's mad cow disease. It's another word. Prions, we don't know for sure how you get infected. So if you eat a meat from mad cow disease, how does it affect your brain? Like, how does it get to that part of you? Like, how? We don't know. It's just a protein. It's completely changed how we see infectious agents. It's not got a nucleic acid or anything. It's just a protein. And it's a protein that we have normally in our bodies. So what is going on there? It seems to be misfolded. It's not folded correctly. If you've ever read Cat's Cradle, by Kurt Vonnegut, then it's similar to Ice 9. It's like Ice 9 was like a concept in that book where it was like water, like preferentially formed crystals to form Ice 9. But it was so dangerous that it would cause all water in the world to want to preferentially interact that way. So they had a crystal, and then a guy like broke open the container of it and like killed the whole world. I think it ends up happening in the end, but like it's this big old thing leading up. It's a lot more involved than that, but you get the idea. It reminds me of Ice 9. So anyways, that's what this is like. They don't have any nucleic acid. So how do they replicate?
There is evidence that the infectious agent associated with certain brain diseases, such as Creutzfeldt-Jakob disease in humans and mad cow disease in animals, may be caused by a protein. The infectious agent has been called a prion, for proteinaceous infectious particle. The normal prion protein, PRPC, is bound to the surfaces of neurons, and the gene for PRP is present in normal vertebrates and invertebrates. Presumably, the normal prion protein, PRPC, can be altered, and the altered prion protein, PRPSC, SC meaning scrapie associated, becomes misfolded and takes on a different conformation. Many researchers believe that the infective agent is this alternative protein conformation. It is believed that when PRPSC enters a normal brain, it binds to the normal cellular PRP, PRPC, and induces it to fold into the abnormal conformation. The newly produced PRPSC molecule may then convert more normal PRPC proteins into the abnormal PRPSC form. An alternative hypothesis is that PRPSC could activate enzymes that modify the PRPC structure, not shown.
All right. So prions or prions, however you want to say it, what are they made of all together? Like everything. Like if you could choose more than one from here, what is everything that they're made of? Well, lipids are part of membranes and fats, right? Carbohydrates are sugars. Nucleic acids are like DNA and RNA. Prions are proteinaceous infectious particles. They're just proteins. That's all there is. Okay. All right, so the other things that can be infectious, but that are not cells. We have satellite viruses. They depend on other viruses for replication. The most probably well-known one is adeno-associated virus, AAV. It was originally being looked at for like gene therapy use. And gene therapy means like if I could like take a virus and get you infected with it, but instead of it like putting its, stupid virus genes in you, it would be putting like the gene to make insulin or something like that. So that's the idea anyways. So originally we thought that AAV, adeno-associated virus, could only infect cells that were infected with adenovirus. Now we found that it can infect cells that have been infected with other viruses or have their DNA disrupted through other means. So like cells that have other problems with their DNA. And then we have viroids. They're just virus-like agents that are in plants, really just in plants. They're just naked RNA. They're just RNA. So that's all it is, but they can cause problems in plants. So totally overall, we have in this chapter 6, talk about viruses. Viruses infect all cells ever. Why is this? Why are you doing that? All cells ever, they consist of a nucleic acid. It can be DNA, it can be RNA, it can be single-stranded, it can be double-stranded. It can be the positive, which carries the message, or it can be the negative, which has to make a copy to carry the message, right? It can be any of that, but it can only be DNA or RNA. It cannot have both going on, right? Your little head's in the way. I'm elbowing you. Okay. I know. Moving on. They have to have a capsid. So those are like the minimum, right? The nucleic acid and the capsid. And the capsid, what does it consist of? Capsomeres, repeating units of capsomeres. And then we have the envelope on the outside. That's the membrane layer taken from the host cell. And then we have the replication cycle that we talked about. We went through each step. Adsorption, the attachment, penetration, getting into the cell, uncoding, dropping off everything, letting its nucleic acid go, synthesis, making copies of the nucleic acid and of the proteins involved, like the capsid and like the spikes, and then assembly, putting everything, all these parts together, and then release either through budding or exocytosis, right? Some viruses can undergo lysogeny or latency. That means incorporating that viral DNA into the host's DNA and just kind of hiding out there. And then prions or prions are proteinaceous infection. They are, that's not a good way to word it. Prions are infectious protein that affect the nervous system, like spongiform encephalopathies. Oh my gosh, yes. Sorry, my husband got home at the end and it scared me. So it really threw me off, but that's it for chapter 6. And that's it for unit 1. So we'll be moving on to unit 2 next. And I'm sure that you guys are using all your study techniques to study and all of that. And I can't wait to see you in unit 2. Stop, Hazel. Jeez.